One of the first things you probably think of when you hear the word mutation is either the Ninja Turtles or the X-Men. And it's true, both are mutants. Something about them has been fundamentally changed so that they have different appearances and abilities that are coded into their DNA. I don't know as much backstory with the X-Men as I do the Ninja Turtles, so I'll use them as an example, so you'll have to forgive me and fill me in on the X-Men. But the Ninja Turtles were exposed to radioactive ooze early in their development, which caused the mutations that they have. Just because these mutations change their DNA doesn't necessarily mean that they'll pass them on to their offspring. And this may be more applicable to the X-Men because the Turtles don't have any children, nor do I foresee that in their storylines. If these mutations affected their gametes, the mutation will be able to be passed down to their children. There are two different categories of mutations we're going to look at in more depth. The first is a gene mutation. In a gene mutation, just one particular gene is affected. Many genetic diseases are gene mutations. Here's an example of sickle cell anemia. The top strand is the DNA sequence, and the strand underneath that is the mRNA, and beneath that are the corresponding amino acids. The codon highlighted, or the DNA codon and amino acids that are highlighted in blue is showing that a mutation has taken place in sickle cell anemia. You'll notice that the T is changed to an A in sickle cell anemia. In a protein of thousands of base pairs, just one single letter is changed to produce the disease sickle cell anemia. This is a disease that primarily affects African Americans and results in their blood cells being crescent-shaped. This makes it harder for oxygen to attach to red blood cells, and it makes it easier for them to get stuck in tiny blood vessels. As a result, people with sickle cell anemia experience blood clots easily and have tremendous pain when their blood cells stick to the vessel walls. Other genetic diseases can be caused by chromosomal mutations, though they're rarer. Chromosomal mutations affect sections of a chromosome which contain many genes, or may change how many chromosomes an individual has. Chromosomal mutations generally have more widespread symptoms because they involve many genes. An example of a chromosomal mutation you are probably familiar with is Down syndrome. Individuals with Down syndrome experience a wide variety of symptoms. Besides the characteristic facial features, people with Down syndrome typically have heart problems, poor muscle tone, mental retardation, intestinal problems, and growth problems. You can see that this causes symptoms throughout the body. The particular type of gene mutation present in sickle cell anemia is called a point mutation. Looking back at the DNA strand between the normal and mutated DNA, we only saw one base difference. Point mutations can be caused by mutagens, which is simply something that causes mutations. UV light can act as a mutagen, changing the DNA of skin cells, which leads to cancer. When you go to the dentist and get x-rays, they put a lead bib over you so that your body is exposed to the least amount of x-rays as possible. That's because x-rays are also mutagens. Rosalind Franklin, who worked with x-rays as a part of her daily work, died of cancer at age 37 because her DNA was mutated by the x-rays. Chemicals and drugs can also act as mutagens. Chromium-4 was the chemical found in the water in the movie Erin Brockovich. You've seen that, which was based on a true story. Essentially, there was a chemical that seeped into the water because of an energy company, and a lot of people were getting sick and getting cancer. And so Erin Brockovich was the sort of detective that figured this all out, even though she was sort of a small-town girl. Mutagens damage DNA, which then in turn gets transcribed to mRNA and then translated to a protein. There are three types of point mutations. Their name describes how the DNA is affected in each case. In a substitution mutation, one DNA base is traded for a different DNA base. As you can see in the first picture, only one amino acid was ultimately affected, and that was the one that contained the substitution mutation. Point, insertion, and deletion mutations are considered frame shift mutations because adding or deleting a nitrogen base changes how the rest of the mRNA sequence is read. It's called shifting the reading frame when the groups of 3 bases are shifted because of a newly added or deleted base. You can see the insertion and deletion have caused all of the amino acids after the mutation to be different. Chromosomal mutations affect the entire chromosome. Whole chunks of the chromosome which contain many genes are impacted. If the original chromosome is broken down into 6 sections, A through F, a deletion is when one of those chunks is deleted. Duplication, like its name suggests, is when a whole section is repeated. Inversion means the order of the segments is changed. In this case, 2 segments are switched in the middle. B and C have changed places. Finally, translocation is when a section from another chromosome, not a homologous chromosome, a totally different one, attaches to the chromosome that it doesn't belong to. The word mutation sort of has a negative connotation to it, but really, most mutations are probably neutral. It's hard to know how many neutral mutations a person might have because they have no effect on us at all. Many can be neutral, though, because the genetic code is redundant. Usually 3 or 4 different codons code for the same amino acid, so changing from one codon that codes for tryptophan to a different codon that also codes for tryptophan makes no difference when the protein is actually produced. Some mutations are bad and result in serious diseases such as sickle cell anemia, Huntington's disease, and cancer. Many of these diseases can be passed down from child to offspring if the gametes are also affected, and so they're called genetic diseases. There are many cases where mutations are actually good. Mutations create genetic diversity, which means that we're all different. In organisms like bacteria, some of these differences might actually be advantageous and allow them to survive longer and reproduce more. Humans have beneficial mutations also, but this generally doesn't lead to us having more children. There are a lot of things humans do to prevent having children. It's also possible for plants to have mutations where they have many duplicated sets of chromosomes. This condition is called polyploidy. It might sound bad because just one extra chromosome in humans can cause serious genetic diseases, but in plants these extra sets are actually good. They give the plants larger fruit and produce plants that can withstand a wider variety of environmental conditions. They can also produce fruits that don't have seeds in them, so seedless grapes actually have many sets of chromosomes. In fact, most of the strawberries you eat actually have 8 copies of each of their chromosomes. So don't forget to go back and take the quiz now that you're finished watching the lecture. So here's a preview of your quiz.